What's going on, everybody? This is Dr. Adam Rindy, and welcome to the next episode of the One Thing podcast. I'm delighted to bring to you my special guest, Scott Anderson, the author of the Psychobiotic Revolution, which is a book on mood, food, and the gut-brain connection. It goes into the concept of psychobiotics, how probiotics and prebiotics may influence brain health and brain function. He co-authored this book with microbiome experts John Cryan and Ted Dinan. In this episode, we go into this topic on a very deep level. The episode picks up mid-conversation, so without further ado, we're going to jump right into the episode. If you like this episode, please make sure to click like on your podcast player and subscribe to our channel so that we can further get this message out. Thank you for tuning in. I greatly appreciate it. Enjoy the episode. If we could start with the hologenome perspective and just kind of describe what you mean by that and how that is kind of, in my opinion, a way to set the stage for today's discussion.
Excellent. Sure. The hologenome idea is that we are always assuming that our genes are the important genes and that we pass them down, and we do. And that is a major aspect of our personality, the way that we handle stress and all of that. But our genes actually constitute about 1% of the entire genetic complement in our body. The other 99% is supplied by bacteria. So there's thousands of species of bacteria, each of which contains hundreds of genes. And those genes are capable of doing amazing things. In particular, this is shocking to a lot of scientists and still kind of blows my mind, is that bacteria can produce human neurotransmitters, including acetylcholine and dopamine, serotonin, things that are typically used by psychiatrists as drugs for dealing with depression and anxiety. So it's fascinating to see that bacteria can produce these. Now, we don't really know how all of that works, and I guess we can come back to that a little bit later. But the idea is that in your gut, there are 100 times more genes than you have in your body. And we have co-evolved with these bacteria over millennia. And the idea of this is kind of striking, but we as humans are pretty good at coming up with changes over long periods of time to our genes, but it can take us a millennia to change a gene, whereas bacteria can change a gene in 20 minutes or so and then produce a flood of new bacteria that have this new gene that they can pick up from other bacteria, or they can pick it up from viruses, and they can even pick up some genes from humans, and vice versa. Humans can actually adopt genes from viruses and bacteria as well. So there's a lot of crosstalk between all of these things, and it means that looking at us as strictly humans is not the way to see how we are going to respond to diseases or stresses in our lives, because we have all these other genes that we need to look at as well. So several of these microbes that exist in our gut exist no place else in nature. They can only survive inside of our gut, which is a fascinating concept in and of itself. And that means that we must treat them well. There is some speculation that we are actually driving certain of these bacteria to extinction by virtue of the fact that we're not treating ourselves well and we're not feeding those bacteria properly. Mm-hmm. So this idea of a hologenome, the idea that we are really the sum of all of the genes, not just the human genes, but the bacterial genes as well, fits the data quite well and lets us know that we should probably be paying a little bit more attention to the health of our microbes.
Mm-hmm. Yeah, it's amazing to think, as you're talking about how the bacteria adapt and will change their genome. It's like, yeah, it makes a lot of sense. Look where they're living. I mean, what a harsh environment.
Right. And it's the evidence seems to say that there's a recent study that came out, I think it was just this year, that said that they found something like another 50% of the genes or the microbes in your gut have been undetected because they have such a small genetic load. And the reason is that they've been able to drop genes. And those genes, when you drop a gene, if you can depend upon the kindness of other microbes, you can survive without these genes. And as a consequence of losing them, you have less energy needs. So it can actually be beneficial to be very small and very lightweight when it comes to genes. But those were bacteria that we haven't detected until recently. So it's possible for these bacteria to pick up new genes and lose genes in a sort of helter-skelter way. It's not at all like the way we think of humans. It's very fluid. And it's turning out that probably what we should be looking at more than the microbes themselves are those genes. Those are the genes that are doing the work. And although they're associated with microbes, as you can tell by the fact that they keep switching genes around, it's probably not as useful to look at specific microbes as it is to look at specific genes.
Interesting. Yeah. So it seems like we go back and forth between, looking at the organism versus looking at the gene level in science.
Right.
Well, you're an expert in brain gut issues. And I think if you could just describe to us, what is the brain gut axis and why isn't it called the gut brain axis? Or maybe it should be. Can you just kind of tease that out for us?
Yeah, in fact, it depends upon whether you're an enterologist, a gastroenterologist, or a neurologist as to whether it's gut brain or brain gut. And this is a field that is involved with immunologists, endocrinologists, psychiatrists, and microbiologists, as well as neurologists. So it's a fascinating field where you have people, there's a lot of intramural sniping, as you might imagine, where microbiologists are wondering what neurologists have to say about bacteria. And neurologists are wondering what microbiologists have to say about the brain. But they have all kind of become a little bit more humble in the face of this new science. And they're accepting that there are a lot of things that everybody has to learn about this. The understanding, the connection between the gut and the brain goes back to Hippocrates, who basically said all disease starts in the gut. And when he was talking about that, he was also talking about diseases of the mind, because he was one of the first to understand that people may have mental issues that are actually based upon some physical substrate in the body. And they didn't really know that it was the brain or all of that. The details weren't there. But because Hippocrates could figure out ways to fix it, he felt that those were not off limits to people who were studying medicine and biology. Back it up, now you come up to the more modern era when bacteria were first discovered. And immediately the connection was made between bacteria and diseases. And that was the germ theory of disease. And the idea was that basically bacteria were all pathogens. And so we set out to just annihilate them. And we did a pretty good job with the invention of antibiotics. We're able to kill a lot of bacteria. But recently we started to figure out, wow, some of these bacteria are actually good. And that was around the time of Louis Pasteur, who figured out that some bacteria were creating wine, and that was good. Other bacteria were making your wine go bad. That was bad. So he's the one who figured out how to get rid of the bad bacteria and enhance the good bacteria. And that was a big breakthrough. And of course, we all wine lovers have owe a big thanks to Louis Pasteur for figuring out how to get that to work. And in our bodies, we've seen the same sort of thing. We've seen one of the first instances where people understood that there was a weird kind of gut bacteria thing going on was something called hepatic encephalopathy. And that's where you have a liver disease, but all of a sudden you start going a little crazy. You don't act like yourself. You get angry with people. You strike out at your relatives, and everybody was dumbfounded. And they found that those people could be helped with a big dose of antibiotics. And that was a little shocking. It's like, wait, what's going on? It wasn't bacteria in the brain. It wasn't bacteria in the liver, although it's partly bacteria in the liver. But it was gut bacteria that was influencing this mental problem. And The reason that the gut is the source of all of this is that there's just so much bacteria in there, all of them presumably working to help you in a lot of ways. But if they get into the blood circulation, none of them are any good.
Mm-hmm.
And your heart gladly pumps them to everything, and it'll pump bacteria into your liver, and that's where you can get some of these encephalopathies. And so it's quite an interesting connection that made people sit up and pay a little bit of attention to that, to what's going on there. And then there was another one in 2000. Walkerton, Canada, had a flood, and it washed a whole bunch of fecal matter from a chicken farm into the water supply.
Mm-hmm.
And they got a bacteria called C. jejuni, and that caused the whole town got sick. Caused the whole town to end up in the hospital. 5,000 people were sick, and several people were killed by this bacteria. And those people got something called post-infectious IBS, irritable bowel syndrome. And some of them came down with depression. A good percentage of them had depression that lasted for quite a while until the certain scientists from McMaster University in Toronto looked at them. That's Stephen Collins and Prinzel Berchek. They went and looked at them, and with drugs, antibiotics and stuff, they were able to help these people both with their IBS and with their depression. And that was one of the first times, again, that was a real eye-opener in terms of how bacteria can affect your gut. Bacteria can affect your brain. And so that was the beginning. What we were seeing, though, in both of these cases is how bacteria can give you a rotten mood, basically can cause depression. And so the bigger breakthrough started to come with John Cryan and Ted Dinan, who realized that some bacteria could improve your mood. And that's where the excitement comes from, and why I decided to do this book was when I looked at that, I just said, that makes perfect sense.
Yeah.
And these guys were, So at any rate, that's where that stands.
Yeah. So it seems like the kind of what you've described is sort of an endotoxin that's coming across from the gut and getting into the systemic circulation or dumping into spaces in the body, like the liver, the brain. And it's, sort of an inflammatory problem, which you talk a lot about in your book, that, depression specifically and maybe anxiety are related to inflammation. Yes,
it seems that inflammation is at the root of probably, at least from what we're seeing, probably 90% of chronic diseases. So we're seeing inflammation being at the root of diabetes and obesity, arthritis, being at the root of, amazingly, being at the root of Alzheimer's and Parkinson's. That is fairly new understanding. The story with Parkinson's is that bacteria start and create something called Lewy bodies that are in the cells that are in the nerve cells that surround the gut. And they work their way up through the vagus nerve. It works its way up. It takes about 10 years to get to the brain. And when it does, it has these Lewy bodies keep spreading up to the substantia nigra, where they can cause Parkinson's. And these Lewy bodies are quite likely to be some kind of a prion type disease. You're looking at proteins that fold differently when they sense bacteria. So it's probably an antibacterial effect that is being done that is causing its own side effects. It's your immune system can cause a lot of damage in its eagerness to clean up bacteria. So that's kind of an interesting way of looking at things. And with Alzheimer's, the association has also been there in particular with things like Porphyromonas gingivitis. That is a bacteria that causes gingivitis in your mouth. But they have found toxins related to that bacteria in the brains of people with Alzheimer's. So for a long time, Alzheimer's has looked at alpha-synuclein and, beta amyloid. Alpha-synuclein is in Parkinson's. Beta amyloid is in the brains of people with Alzheimer's. And that has been the subject of a whole bunch of drugs that have been trying to get rid of the beta amyloid. And getting rid of the beta amyloid has not worked. 400 drugs have been tested that get rid of beta amyloid.
Mm-hmm.
None of them stop the progression of the disease. And that's likely because the beta amyloid, to use a fire starter metaphor, the amyloid is the fireman. It's not the arson.
Mm-hmm.
It's not the arsonist.
Mm-hmm.
And we've been treating it improperly that way. So the actual arsonist would be the bacteria that have managed to get past the blood-brain barrier. And then the amyloid is the body's defense mechanism trying to trap them. So getting rid of the fireman is not helping the arson situation in your brain. So inflammation of that kind, any time you have what they call a translocation of bacteria across the gut barrier, is problematic. But it doesn't have to be, actually, it doesn't have to be whole bacteria. Just pieces of bacteria can also trigger inflammation.
Mm-hmm.
So that's another thing to look out. Lipopolysaccharide, LPS, is something that gets across pretty easily, and that can trigger a pretty strong inflammatory response.
Right.
So all of these things, these are infections that get into various parts of your body, and the inflammation itself can be, it's what you need to do. Your immune system needs to go after them, but sometimes it can't get rid of them. The infection is ongoing. The gut is permanently leaky, and so you've always got these bacteria going in. And because the immune system is imperfect, it can sometimes attack you as well. Attack your own cells. And sometimes that's the only way to deal with an infection. An infection that's inside of a cell, the way that you deal with it is to kill the cell.
Mm-hmm.
So there are infections, there are bacteria between cells, and there's bacteria in cells. And those are different, inflammatory modes. And how the body deals with them is not necessarily the same.
Yeah.
And so these ongoing inflammations, that's really problematic. And that's probably what is behind almost all the diseases that plague us outside of, say, infectious diseases.
Okay. So I want to pivot on that because I think that's a really good point. So you describe these two different types of mice that are used in studying the microbiome. A mice that has maybe very little bacteria, then a mice that has no bacteria in the gut. And can you just kind of tell us a few things, key points that we've learned about these two different species of mice and how that's really influenced how we see this gut-brain axis?
Yeah, the early studies with germ-free, they call them germ-free mice. They raise these mice, they deliver the babies through a sterile C-section and raise them in a completely germ-free environment. So they basically have no bacteria that we know of. Now, they may still have phages, they may have viruses. We don't know for sure. It's not sure that it's completely sterile, but as far as we can tell, there are no bacteria with these mice. And this was a study done by Nobuyuki Sudo in 2003, I believe. And he started out with these bacteria-free mice. They're in this kind of funky environment where you have to reach in with sterile gloves and stuff to feed them. The food has to be all sterile and everything. And those mice are different. They expected to see some physical differences, but what they found was behavioral differences, and that was striking. And in particular, they found that they responded differently to stress, and that these mice were, when you stress them, they had an overreaction to the stress. In other ways, they were a little bit more adventurous, but in some ways, they were kind of like antisocial. So it was different. The behavior was different. And they realized that if they gave them some bacteria, kind of in pathogen-free bacteria, that they could restore their behavior to normal. And that those mice, then the mice that have specific bacteria that we know about, and usually a small number that's tractable to deal with, say a dozen or so bacteria, different genre of bacteria. Those mice are called autobiotic. They are not germ-free. They have germs, but they have very specified germs, and they're free of pathogens. And so with those two different types of mice, they were able to find out it's hard to know when you have 1,000 different genera and species of bacteria in your gut. It's hard to know which one is important and which one is calling the shots, if any. And so you're looking at a vast and complex ecosystem in your gut. So bringing this down to zero in the case of germ-free, or just a dozen microbes, microbial species in the case of not-abiotic mice, the whole problem becomes a lot more tractable. You can start to see networks of bacteria and how they work together to create what they call homeostasis in the gut. And when that gets unbalanced, when one species starts to take over, that's called dysbiosis, although that term is kind of losing its cachet because it's hard to nail down exactly what dysbiosis means.
Mm-hmm. So that was sort of the launch of psychobiotics when we started seeing these patterns? Okay.
Yes, that was really when it started out with a vengeance. All of a sudden, everybody's saying, well, wait a minute. So you're saying that if you don't have bacteria, your brain is different? They looked at their brains and they saw that the development of the brains were different, that they had different rates of myelination around the neurons, and that they had smaller hippocampuses. So those are hippocampi. That's the part of a little horseshoe-shaped organ in your brain where memories are initially stored before they go to long-term memory. And so the hippocampus is quite an interesting organ. In London, taxi drivers who have to know all the streets in London have giant hippocampuses. And that is a great story for people to realize that the shape of your brain, the size of different parts of your brain can change depending upon your environment. Mm-hmm. And they can also change and be different depending upon your microbiota. So this is another thing from Kirsten Tillich is that she figured out using fMRI that certain parts of the brain were bigger. The amygdala was larger in some of these women, and the hippocampuses were smaller in some of these women. Huh. And wait. These are women that yeah, go ahead.
Yeah, you said something that just really surprised me. So taxi drivers in London have larger hippocampus regions in their brain. Yes. So what's that all about?
Apparently, the idea that they have to memorize so many streets causes them to have more nerves generated to accommodate these extra memories. So it's an interesting factoid. It's one of those things that's great at the cocktail party, I guess, if it's a cocktail party full of nerds. And so it's one of those interesting factoids that has led us to understand a little bit more about how the hippocampus works. It's not clear, or hasn't been clear historically, how much a human brain is going to create new nerve cells. The dogma for a long time was that we don't get new nerve cells, but we've since understood that we do. And in particular, in the hippocampus, new nerve cells are being generated at a pretty hefty rate. And that is going to cause the hippocampus to get a little bit bigger. Nerve cells aren't very big. It would take millions of them to appreciably increase the size, but that's actually what happens in these taxi drivers. And they just have all this, they have this map in their head, in their hippocampus of London. And that is how they ended up with extra numbers of neurons. We see the same thing in people who have to learn a lot of things and memorize them. Mm-hmm. So it's something that means it ties the hippocampus directly to memory. So when you see a somewhat atrophied hippocampus in a mouse, you know that it's affecting its memory. Right. And that's one of the things that we saw in germ-free mice, is that you see that they have an atrophied hippocampus. And if you give them a new microbe, a new microbiota, you can rescue that, and their hippocampus will grow to a normal size.
Okay. Well, yeah, and we're going to talk about later on just a little bit about FMT, the fecal microbial transplant, and I want to get into that a little bit. If you were, say, a listener or a clinician out there, and you were just wondering if your brain was affected by your gut, and let's say someone has some anxiety or depression, or maybe they have a condition like fibromyalgia or chronic fatigue, or maybe they're having their early onset of dementia, and they were just to sit back and say, I wonder if my gut is a problem. What would be some next steps that you've seen from the research and just from having higher level conversations as to the best way to find out if the gut is driving it, or do we even know?
Yeah, well, we do know, and more and more all the time. First of all, if you look at gut issues like IBS and Crohn's and ulcerative colitis, these things are highly comorbid with depression. And that's one of the first clues that depression is related to what's going on in your gut. And if you can resolve those things, if you can resolve IBS, which isn't always easy, of course, if you can resolve ulcerative colitis using various drugs, you can improve the depression in these patients. So when you come into a psychiatrist, there are lots of kinds of depression, of course. You can be bereaving, you can be in grief. And there are financial reasons to cause people to get depressed. But if you can't really find anything in their life that is causing it, you should probably look to their gut and ask them, do you have issues with your gut? Or do you have IBS? Do you have gassy problems? Do you have problems with diarrhea or constipation? The whole concept of Parkinson's starting in the gut actually is interesting because it starts with constipation. And most people with Parkinson's have constipation, and they have had it for about 10 years, the amount of time it takes for the Lewy bodies to move up the vagus nerve. And so these are questions that are not usually asked, and they should be asked. Besides that, we can now do a whole bunch of tests to show if there's low-level inflammation. And in general, when you find inflammation of any sort in the body, the most likely source is the gut, because that's this big kind of very fragile ecosystem where you're supposed to be able to absorb nutrients, but keeps bacteria out. So you've got this kind of a tough job of letting things in and keeping other things out. It's not easy to do. And when it breaks down, bacteria can get into your system. So the things that people can look for in the blood are things like C-reactive protein. And there are other things. Zonulin is something that is a part of the tight junctions that make your cells stick together in your gut. And if you see elevated levels of zonulin or C-reactive protein or several other blood signs of inflammation, that's a really good sign that you've got problems with your gut. And that's also a good thing to look at in terms of depression and anxiety, because those things are, to some extent, they are driven by these inflammations.
Right. And what goes along with systemic inflammatory markers like CRP is cytokine markers that they don't traditionally offer like in a standard lab, but that's what the researchers use, the different interleukin forms that are associated with depression and anxiety.
Exactly. You've got these numbered interleukins that are, some of them are good for you, that are anti-inflammatory, and some of them are pro-inflammatory. And by looking at those, you can get a really good profile of exactly, it'll help you know whether you got parasites or whether you have bacteria, or whether you have phages. Different things will cause different parts of those cytokine systems to light up. So that's an excellent observation, Adam. That's one of the things that we do in our own research.
Okay. And what do you think about the DNA sequencing for stool testing? Is that a useful tool now that they're commercially available?
Yeah, there's some issues right now with some of the companies themselves. Ubiome seems to be blowing up right now. I still think that their basic research is sound, and I think that there's something to be learned there. But it's hard to know exactly right now. If you look at studies that are out there, they're going to say the Firmicutes to Bacteroides, these are two major genera of bacteria, that ratio is supposed to tell you something. But every single study that I look at has a different value. And sometimes the ratio is greater than 1. Sometimes that ratio is less than 1. And sometimes that's supposed to be positive. Sometimes that's supposed to be negative in impact. And so right now, I think that that is insufficient to really tell you whether you're sick or not in terms of your gut balance. And again, this also comes back to the idea that these bacteria themselves are hard to pin down, knowing that, because how do you know what is a bacterial species? Well, you look at its genes. Oh, they're swapping their genes. Well, that kind of blurs the lines between microbes in the first place. So it's kind of tricky to do that analysis and come away with a feeling that you really nailed it. A way to look at this is in terms of diversity. And this comes back to an ecological point of view about how your gut works, is that a greater diversity in general seems to imply greater homeostasis. It implies that not one species or one or two species have taken over, but that everything is fairly well balanced. When you have less diversity, you tend to have a few species that are taking over, and that are kind of being bully species, that are now out more for their own sake than they are for the sake of the team, let's call it. And so that's something that would be nice if we could get that from the fecal transplants, or from the fecal analysis. It's hard to see that that's what you're getting right now. And again, this is one of those things where some people can look at this and get some really good results. The folks at the Weissman Institute are using fecal analysis, and they're getting better results. And then the new way of doing fecal analysis is not with I better backtrack for just a second. The first way that we used to look at fecal genomes, so the fecal, microbiota, was to culture them. And not many of those bacteria could be cultured. We've gotten better at culturing, but we've gotten even better at looking at them using DNA analysis. And the DNA analysis that we use is to look at a single part of a single gene that corresponds to the ribosomes. All of these creatures have ribosomes that are little factories that produce proteins from the genetic blueprint. And that is pretty much conserved from one species to another. So if you look at that, it's called 16S rRNA. That is what most of these companies like Ubiome and a lot of the papers that we've seen over the last 20 years or so have been using that to identify bacteria. But they can only take it down to the genus level. They can't really get it, very rarely can they get it to the species level. And that has been helpful, but not as helpful as a new form of analysis called whole genome shotgun sequencing. And that looks at every single gene that's in the sample. And from that, they can piece together not only what genes are there, but what bacteria are there down to the species and even subspecies level. And they can find other things as well. They can find parasites and they can find viruses as well. So these are new techniques that are being used, and that's one of the techniques that was used in that large Belgian study that I mentioned earlier. And that's one of the ways that they were able to nail down so much more in terms of species than any other study that has been done.
Mm-hmm. Yeah. So I think, it seems like we're, a few of the clinical or commercially available tests will look for pathogens and then look for potentially what are called dysbiotic organisms, and potentially look at the good, quote, good bacteria that we want to be rich in our gut. So I think that's kind of where we're at right now, is but it's interesting to see when you talk about microbiome diversity. Could you just briefly talk about some of the terms that we might think about, like the richness of the gut microbiome and how that's even graded?
Yeah, there are a lot of different ways of looking at richness. The first, statistically speaking, this is a ripe field, and I think that it's ripe also for some upgrading. But we look at richness, just the simplest one is just how many species do you have? How many different species do you have? But another way of looking at it is to find out what is the actual numbers. And we're not seeing that in most of these studies. You don't see exactly quantitatively how many of a certain species you have. You may see relative amounts, and that's another way of looking at it. But it would be nice to know exactly how many of each of these bacteria that you have in your gut. And you put a quote around pathogens, and I think that's appropriate too, because things that are pathogenic in a situation of dysbiosis may be perfectly reasonable citizens of a normal microbiome if they are at the reasonable numbers. In other words, Clostridia, C. diff, for instance, Clostridia difficile, is a well-known pathogen that can kill people, especially people who've had antibiotic treatments. But in your body, Clostridia is some of the more helpful bacteria in a normally balanced gut. And they are the ones that produce butyrate. And that is something that is like ambrosia to the cells lining your gut. So Clostridia species are actually very beneficial if they're in the proper ratios and if they are compared with other bacteria, not dominating.
Right.
And so that's a big issue. And it means that there's not really such a thing as a commensal bacteria or a pathogen, at least in your gut, because it all boils down to how well they're playing with the other bacteria that are down there.
Yeah. And, I think that's one of the things I speak a lot with my patients is, it is very host dependent. If something comes back on one of these panels, it could be living, you could be managing the inflammation or potential inflammation that particular bug is causing in your body versus the next person might be having problems handling it. And so it gets very tricky. So I think when people get these panels back, it's important to take a step back and say, okay, what is my body actually doing? Am I having signs and symptoms of someone who has a C. diff infection, which those symptoms usually are pretty extreme on the scale of most digestive disorders. So yeah, I think it's a really good thing for us to kind of continually revisit is that there's the bacteria, there's the host, there's the interrelationship between the bacteria and other bacteria, then there's the relationship between the host and the bacteria. So it takes a lot of art to figure out the significance of these panels.
Yes, art is correct in this case. And that's, in a lot of science, what happens is it starts with art and then we get a little bit better with the numbers on it and we can nail things down better. But you're absolutely right. It is fantastically complicated because your genes will interact with the bacteria in your gut. And what's going on sort of from a bigger evolutionary perspective is that you got your gut bacteria largely from your mother, and at least in most cases we do. We get it as we come down the birth canal. We pick up some bacteria. We pick up bacteria actually from our mother's fecal matter too, because birthing is often messy. And then we pick up bacteria from breastfeeding. Mother's milk is full of bacteria. And those are bacteria that your mother that is passing down to you from her mother and from her mother before her. And those are bacteria that work well in her system, given her specific genetic proclivities. And we find that a lot of these diseases that have genetic components, those genetic components are largely in the immune system genes, the genes that control immunity and how they react to the bacteria that's in your gut. So this is a huge, complicated affair, very data intensive when we try to deal with it. And it's causing people to scratch their heads who are used to dealing with large databases, but all of a sudden they're realizing, the human database, the human genome, which is large and perplexing and hard to deal with the data, with even the biggest computers, now multiply that by 100 because you've missed most of the genes. And so it is really the interaction of all of those genes that is causing your particular situation. And everybody is going to be unique. And that's a really, it's a big wake-up call for the whole business, is to realize that there is no one-size-fits-all answer to these things. And that's why in my book, I'm really encouraging people to keep notes about what they do, what works for them, and then to go with what works for them, and kind of not to ignore what's going on with everybody else, but don't try and compare yourself with somebody else who's had a fantastic time with lactobacillus or something. If it doesn't work for you, there might be reasons why it doesn't work for you.
Right. Good point. Yeah. So, well, I'm going to move from this excellent conversation and background information to kind of moving towards applications and just some sort of practical suggestions.
Yeah.
So you have, there's some key players in the microbiome, I guess we could call them probiotic species, that I'd like to just kind of hear a few comments on, because these are when people are going out and looking for probiotics, they're familiar with some of these terms. So if we could just talk about these and how they relate to brain health, like first of all, maybe some of the lactobacilli species, and then go into bifidobacteria, and then I'd like to hear your thoughts on the Akkermansia.
Oh, right. Okay, well, actually, let's start with bifidobacteria, because that's the one that you start with. That's one of the first ones that's in your mother's milk that started to populate your gut. And as you get older, you get less and less bifidobacteria, which is really dedicated to dealing with milk and milk sugars, and goes on to lower numbers as you get older. Bifidobacteria, though, is very important and is in things like yogurt, as well as lactobacillus, which is another one that starts you out early. Lacto, obviously referring to milk as well. And so you're looking at a couple of bacteria there that will start you out early and let you take the maximum advantage of mother's milk. And then as you get older, sometimes the lack of those bacteria is what leads to things like lactose intolerance and stuff. So there are changes in your gut bacteria as you grow, and trying to bring some of them back may work. It may not always work. Different people show different results. Yogurt is one of those things that contains large quantities of lactobacillus and bifidobacter species. And so that's one of the things that a lot of people have really good luck with in terms of probiotics. You can buy probiotics, you can buy supplements. Some of them are good. In my book, I talk about which ones have actually been studied. There are not that many that have actually been studied, and some of them, not all of them, but some of them are less than honorable. Some of these companies are less than honorable in terms of what they're putting into their products. So you've got to be a little bit careful about that. It's kind of a wild west right now. The FDA treats it as food and doesn't really look at it. And unless people die or something, they will not come and check out some of these manufacturers. On the other hand, there are some good manufacturers out there, that are prominent manufacturers like Procter and Gamble that do produce things, Culturelle and a few other products that are better studied. And so in our book, we try to steer you a little bit more toward the ones that we trust a little bit more, that have actual randomized controlled studies behind them.
Okay. So let's take one of those, like VSL number 3, I think is one that you talk about in your book. Why would that be used in sort of anxiety and depression? Like what's the plausible mechanism of the species that are in that particular probiotic?
Yeah, I don't think there are a couple of things that are going on here. First of all, some of these bacteria do produce butyrate, and butyrate, as we've mentioned before, it's a short chain fatty acid that is actually a primary food of the cells lining the gut. Butyrate also helps to heal those tissues as well if they get ripped, and there's a lot of wear and tear on your gut lining, and so a big part of the action of these things is to maintain good gut health. Other parts of it may be involved with those neurotransmitters we talked about earlier. If you've got bacteria that are producing dopamine, then that can go a long way towards soothing your brain. Although it's not clear yet exactly how neurotransmitters in the gut are sent up to the brain, and this is a place where there's still lots of study that needs to be done. The other thing that's happening, though, and this may be even more important than anything else, is that VSL3, for instance, has a large number of CFUs, the colony-forming units. It has a large number of bacteria in it. Those doses are higher than most any of the other probiotics that are out there. It's also one of the few that has more studies behind it than the others. So it just in terms of sheer numbers, VSL3 may be one of the few that we would recommend, and that is definitely going to have an impact on your gut, as opposed to many of these others that are just going to slide right through without making any kind of impact at all. I tried a whole bunch of prebiotics, probiotics, and prebiotics when I wrote the book, and found very few of them did anything at all. So the numbers of them are probably helping to increase diversity in your gut. And that may be one of the secrets behind a lot of them, is that they're just making things more stable, more homeostatic, and less dysbiotic, so that you can keep the bacteria in your gut where they belong. Keep in mind, though, that your body does not look like a yogurt maker. Yogurt is going to pass through. It's not going to stick. The bacteria in yogurt are not going to stick around and colonize your gut. There may be a few that will, but by and large, they are traveling. They're just traveling through, and you need to keep. While they travel through, they can do good things. They can produce butyrate. They can produce neurotransmitters. They can produce hormones that are beneficial, but they're not going to stay there. And that's why you need to keep eating yogurt. You can't just eat it one day and then say, done.
Right.
Yeah.
And I know we'll talk about prebiotics and maybe some of the more potentially longer lasting treatments that might have like more of a solid non-transitory impact on your gut microbiome in a second. There's one kind of, I guess, the early psychobiotic blend called the probiotic, and it has the lactobacillus acidophilus R52 and the bifidobacterium R175. And so that was used for stress and anxiety in rats, I believe. Any comments on that particular blend and why?
Well, that particular blend, as I understand it, that's not that easy to get in the first place. I don't think you can get it in the United States. But the Canadian, it's a Canadian product, and it's the Canadian government has actually anointed it with its blessing as a genuine psychobiotic. Why it works is still not clear, and the studies with humans are mixed. Some of the things like Bifidobacter longum have been shown to help with anxiety, but not with depression. So there are some issues that are a little bit confusing in terms of what we're seeing, and a lot of these studies came from mice, and there are specific bacteria that work well in mice. One is Bacteroides fragilis, but that is actually potentially pathogenic in humans. So we need to be careful about the what they call the psychopharmacological bridge between animals. We cannot assume that what works in a mouse will work equally well in a human. Although there are some really good studies that show that we can, at least in broad strokes, we can trust the connection, and that's where you get into the fecal transplants. Transplanting fecal material from a depressed human to a germ-free mouse can cause that mouse to act as if it were depressed. So that not only shows that it crosses the species boundary, but it also shows causality. That means that this is not just an associational study. We now, you give them that, and that happens. So
that's amazing.
Yes. So those are studies that give us those are some of the first studies that made me say, "Oh, this is definitely real. This is not just some strange association that could be explained many other ways."
Yeah, just so for the listeners, like if you're getting if you're going in for a fecal microbial transplant, there's a whole donor selection process, and you're screened for pathogens and blatant infectious organisms. Now I understand people are being screened for IBS, patterns of IBS before being accepted as a donor. And now this is a third element to look out for, which is the mental health of the donor.
Right. And it definitely something that you want to look out for is this a cheerful person And also obesity. We're looking at that too. There have been, at least anecdotally, there have been cases where people have had fecal transplants from an overweight person, and they immediately became overweight within just weeks. Their weight started to balloon, and that is perhaps related to the fact that certain bacteria are more efficient than others, and the efficient ones will just eke out the last calorie in any food that you've got. Whereas the slackers are the ones that we probably want more of, because they'll let a lot of things go through and will not help you put on weight. And in that category is Akkermansia, which you mentioned earlier. Akkermansia muciniphilus is the mucus lover. Will actually eat your own mucus if you don't feed it properly. But it is associated with lean people, and it's likely because it's not very efficient at converting food into calories. So these are things that if you are getting a fecal transplant because you're dying of C. diff, these things are less of a trauma to you than just surviving this episode.
Right.
But they are still things that you want to be thinking about.
Yeah, that's a very good point, because fecal microbial transplant has moved into research in other conditions besides C. diff, like inflammatory bowel disease, IBS, autism spectrum disorders. So it's, I mean, I think as the more acute the problem is, maybe it's kind of like a background issue, versus if it's a chronic problem, you might need to really weigh in some of these other factors. And
and so far the results are showing that fecal transplants seem to have some longevity to them, at least for a couple of years or more in some instances. It depends, I suppose, on what problem you're trying to solve.
Right.
But it comes back to this idea that we talked about before, in terms of how are your human genes, going to react to these new microbes in your gut. And so there may be some issues where it's not going to work as well long term. But right now, the fecal transplants are a marvelous therapy, a fantastic treatment that with up to 95% success rate. Unfortunately, the FDA has decided that fecal matter somehow is a drug. And so they are being pretty tough on them. And one person has died from a fecal transplant, and the FDA put a stop to it. So for now, anyway, they have an abundance of caution, and kind of maybe not a really well-balanced view of this, because so many people's lives have been saved. One person died. And they put a stop to it. So that's something where we just have to keep an eye on this and make sure that the FDA knows what we think about this as people who are doctors. They have a voice, and they should write to the FDA and say, look, this is something that we still need to be able to have in our armamentarium. This is a fantastic tool.
Yeah, especially since people are some people are out there doing this kind of like back alley kind of transplants. It's
my advice to your listeners is don't do that.
Yeah,
you would not. It's very much like an organ transplant, and you wouldn't do a heart transplant in your bathroom. So just give that a pass and go to the doctor and figure out a way to get it done. Even if the FDA is clamping down on it, your doctor should be able to get a waiver for you, if you've got a good reason for it.
Yeah. Well, let's dive into dietary measures. There's just a couple things that I think this is a good place for us to kind of last segment of the talk is you talk about the psychobiotic food pyramid. I want to kind of hear your thoughts on that. I also just want to talk about some of these dietary moves that seem to have an influence on the microbiome, such as intermittent fasting, ketogenic diet, or potentially like a vegan high fiber diet. If you could just kind of pepper in a few thoughts about that, and then I want to just kind of hear, like, to leave our listeners with thoughts about the psychobiotic food pyramid.
Yeah, the idea of our problem, the fact that we have such a high incidence of depression and anxiety in the world right now. The WHO, the World Health Organization, rates it as the top disability in the world. And the reason that that's happening, it seems to be related to our processed foods that started to really take off in the 1950s. Made everything easier for people and made really delicious foods like Cheetos and donuts and all of this stuff that are easy to buy, cheap to buy, convenient and tasty. But in order to do all of that, they took out the fiber. The fiber was the thing that kind of made it coarse and not refined white cakes and all of that stuff. So it was a decision that we made. And along with that decision, we also decided to get rid of fat in our diet based upon an old study by Ansel Keys in the 1940s or 50s, who said that that fat was bad for us. And when we took out the fat may be bad for us, but when we took it out, we replaced it with sugar, and sugar is definitely not good for us. And as days go by, we find out more and more about why, but at least one of the latest studies says that sugar gets rid of a colonization factor. It basically stops certain bacteria from reproducing. And those bacteria are, unfortunately, the good bacteria like bifidobacteria. So when we did this to ourselves, and we've done it to ourselves, we have nobody else to blame. We took out the fiber without knowing what we were doing. And it turns out that the fiber that are eaten by, say, hunter-gatherers that still live in Africa, the Hadza and some other people that live out in a kind of a more traditional, older type lifestyle that we, not that long ago, evolutionarily, we participated in too. To the Hadza, the fast food is a gazelle because they are fast and you have to run after them. And so there's a lot of running involved. There's a lot of exercise involved. There's very small amounts of meat, really. Most of it is coming from people who are standing back from the hunt and are digging up roots and whatnot. And their levels of fiber consumption are double, triple, even quintuple the amount of fiber that we eat today. So what is fiber? Fiber is a oligosaccharide. Oligosaccharides are just, the saccharide gives you the clue that it's a sugar, but it's oligos mean that they are several sugars stuck together into a chain. And those are not absorbable by our own intestines. So they pass straight through the small intestines and make it through to the colon, where they are food for bacteria. The bacteria just love these oligosaccharides. And depending upon what kind they are, different species will love them. And depending upon what the pH is, that will tell you what kind of bacteria are going to use what kind of oligosaccharides. And the pH changes as you go through your colon from about pH of 6 or so to a normal pH of 7 toward the anus. And so that change in pH and those oligosaccharides of various types are what help you establish an ecosystem in your gut. So getting the fiber back into your diet is extremely important, and probably the number one recommendation that we have for how to fix this mess that we're in, and to reestablish a normal ecology of gut. So the foods that have higher fibers include artichokes, Jerusalem artichokes and regular artichokes, asparagus, leeks, onions. These are all good foods. The cruciform vegetables like broccoli and stuff are good. It's also good to get these phytochemicals in there from colorful foods, especially leafy foods. And if it sounds like I'm describing a vegan diet, I sort of am. The idea is that vegan or vegetarian diets, from the point of view of your gut, are probably superior. But there is also a need for anti-inflammatory omega-3s, and that's easier to get from things like fish. So what you end up with is a diet that's kind of vegetarian, kind of basically Mediterranean. And that's the diet that is associated with the best mental health. So something along the lines of get a little bit of meat, and especially fish, and largely a plant-based diet will go a long way toward establishing a gut microbiota that will help you feel much better and improve your mood.
Okay. So yeah, it was interesting in your book. I was looking at some of the traditional diets that you were talking about, and
Yeah, yeah.
if you were to kind of blend three model diets, the Okinawan diet, the Mediterranean diet, and the Scandinavian diet, seem to have and blend them all together. Not that I'm suggesting that, but if you look at that integration of the different elements and strengths of those diets, that really is helpful for the microbiota.
Yes.
So
And all of those diets include fish and as well as vegetables. And the interesting thing, though, I think I point this out in the book, is that these people have had thousands of years to come up with their diet based upon their indigenous vegetation and whatever meat that they can find locally. And so they have come up with something that works well for them. Everybody moved to the United States and brought along a little bit of this and a little bit of that, we did kind of blend it all up, but we also blended it with hefty Germanic sausage and all of this stuff. And we haven't had the thousands of years to figure out which of these things fit together best.
Mm-hmm.
And so that's why a lot of people have settled, just, keep it easy, go for a Mediterranean style diet. They've got a lot of veggies, they got a little bit of fish, they got some nuts, and a little bit of wine. And I think wine apparently helps to make your gut more permeable. And permeability is something that you want for nutrition, but you do not want it to be permeable enough to let bacteria through. And it could be that wine is doing that, at least consumed in the kind of quantities that a lot of anxious Americans who are self-medicating are using it. They may be overusing it to the extent that they are actually making a leaky gut. So some small amounts of wine have been shown to be good, although you have to be careful to notice that a lot of those studies are paid for by the wine industry. And so although small amounts seem to have a beneficial effect, it's one of those U-shaped curves where teetotalers are worse off and heavy drinkers are worse off. And right there in the sweet spot with 1 or 2 glasses a night is where the current recommendations are for wine consumption. But it's one of those things that if you can't control it, you should probably throttle back, because it is not necessarily doing your gut any favors.
Mm-hmm. Yeah, I think there's aspects of what wine provides that we can get in our diet. So like polyphenols, we can get them from berries and olive oil, and so I think resveratrol, if that's why people are drinking wine, is also available in supplement form. So these are all good points that you're bringing up, and good things to think about. I know there's a researcher out of I believe he's in Israel, Lerner, who does a lot of studying on intestinal permeability, and he definitely brings up the harms of alcohol on the gut lining. So
yes,
it's a good
yeah.
thing.
So you're right. Yeah, resveratrol and some of those other polyphenols are definitely indicated, but the alcohol itself may not be such a great thing.
Sure.
So, and of course, that's the reason we're drinking it. We're not drinking it for the polyphenols. We're drinking it to get a little relaxation at night. And it's one of the things that is very popular in terms of dealing with depression and anxiety, is that we self-medicate. And that's one of the things that we do the most. A drink at night helps you take the rough edges off of a tough day dealing with a tyrannical boss. So I'm not saying don't do it, but just watch it. And if it seems like you're feeling sick because of it, just throttle back.
Yeah. So I think that kind of brings us full circle, because if you're, if the topic of today is how to address your mental health through good gut health, and I think you really did a great job of kind of walking us through all the different aspects of that, and also talking about how the richness of our gut microbiome may influence inflammation in our body, which may influence our mental health. And so this has been excellent. Is there a take-home message you'd like to give us? And then also, I'd like to hear about what you're up to professionally, and if you could tell us a little bit more about if people, how to follow you. I know you're pretty active on Twitter, and also I want to encourage everybody to run out and get your book, and just if you could talk about anything else you're up to.
Yeah, I'm totally in favor of everybody running out and getting the book. The book is called The Psychobiotic Revolution. It's by Scott Anderson, and it's with John Cryan and Ted Dinan, who are two dynamic geniuses who have really done a lot in the world of the gut-brain axis. Ted Dinan is the guy who came up with the word psychobiotic to describe probiotics that also can improve your mood. So these guys are some of the tops in the world, and the book is out from National Geographic. You can get it at Amazon or any other bookstore. So I encourage you to look at it. There's a lot of detail in there. There's like 500 references in the book, so it's very well researched. And there are just what we talked about today is just a sampling of what's in the book. You can also find me, as you mentioned, on Twitter at Psychobiotic and on Twitter, and that's we've got 14,000 people there who are mostly professionals, but all people are encouraged to join up and learn about the latest in research. I'm posting things from researchers around the world who are coming up daily with new ideas about the gut-brain axis and how psychobiotics can improve our mood. The take-home, I think, for all of this is that we need to understand not all depression comes from the gut, but the stuff that does come from the gut can be treated and can be dealt with. And although you can take probiotics and they can have some effect with some people, you can take prebiotics, which are the fiber, and it comes in pill form. You can actually get powders and pills that contain oligosaccharides, galacto-oligosaccharide, fructo-oligosaccharide. These are some of the things that are out there. They're called GOS and FOS to make it a little bit less tedious. You can eat fermented foods, and we encourage people to try things like yogurt and sauerkraut and kimchi and kefir. Those things are loaded up with lots of beneficial bacteria and can do you a lot of good. I think, though, that our favorite recommendation, we have two of them. One is exercise. Exercise can do an alarming amount of good to your microbiome, can help your gut lining, and as a consequence has shown great efficacy in dealing with depression and anxiety. So we really recommend, if exercise were a pill, you'd pay $1 million for it. And once you get into it, it's not nearly as bad as it sounds. And the other thing is diet, and we recommend diets like the Nordic diet or the Okinawan diet, or in particular the Mediterranean diet, because they are largely vegetable-based, where you get a lot of the fiber that we're talking about, the fiber that will target your microbiota and will increase the amount of healthy neurotransmitters, hormones, and also the short-chain fatty acids that are so good for your gut.
Yeah. Well, wonderful. Well, I thank you for really putting this out there in the world. It's really helped a lot of my patients, and to have a reference to go deeper into about the brain-gut axis and to understand why treating the gut can have such a profound impact on our overall health. I thank you for really putting this out there and kind of launching into this further for not only me, but all my patients and colleagues.
Thank you, Adam. It's been great talking to you.
Okay. Well, you have a great day, and we look forward to keeping in touch with you.
Sure. Anytime, give me a call and we can continue this story because it just keeps growing.
Okay. Thank you, Scott. Take care.
You bet. Bye-bye.
Okay. Bye-bye. Hey, this is Dr. Rennie, just giving you some takeaways from today's episode. I hope you enjoyed it. There's a few tools that we use clinically to help with brain-gut measurement. One is the organic acid urine test. You might look that up There's a couple offered out there. Great Plains Diagnostics and Genova Diagnostics are two of the companies that do offer this particular tool. I also think measuring plasma neurotransmitters is useful. We do measure neurotransmitters such as dopamine, norepinephrine, serotonin metabolites, GABA metabolites, and we can do some of these tests with plasma. We can do them with urine as well. However, plasma seems to be maybe the most accurate. And some of these tools may help with just assessing how the brain and the gut is currently relating from a neurotransmitter standpoint. I also recommend people looking into nutrigenomics. This is using gene data to look at how your gene interrelates with nutrition and biochemistry. And there's a lot of tools out there. I particularly use Opus 23 in my practice. But I thought, people should know that there's some ways to kind of dig in and look at how the brain and the gut is interacting. Heart rate variability measurement is a good way of assessing vagus nerve function, which is a connection and sort of a metrics for measuring brain-gut function. And we're going to be learning more about how these probiotics can influence mood. And this was a great episode. They're printing out the second edition, so I imagine we will be receiving more and more research on this end as the years go forward. There obviously needs to be some other alternatives and adjuncts to improving mental health beyond antidepressants. So this is a hopeful and inspiring area of health, and we'll look forward to seeing you down the road on the next episode. Please like this episode and share it with your friends and loved ones, and let's get the word out on some of these other tools so people become aware, and also so that we can highlight some of these amazing people out in the world that are trying to treat the root cause of illness. Take care.